very good morning. I'm Mooi from Universiti Tunku Abdul Rahman. Today, I would like to highlight the medicinal benefits of Maslinic acids, a pure compound isolated from a local plant named Coleus tuberosus span, or locally known as Ubi camellia. Before I begin, let me share a little bit of my research journey. As young as I can remember, when I was at the age of 7 to 8, I was always requested by my grandmother and mother to search for medicinal plants. Those plants were given to the ladies that approached my grandmother and mother for their health problems. While searching for the medicinal plants, I can't stop asking myself why these medicinal plants have such miracle healing power. The curiosity that I had have driven me to look for answers for my questions. After reading the information recorded in these ancestral prescriptions written by my grandfather, I was inspired by my grandfather's kindness and generosity in helping the needy to treat their sickness and amazed by his knowledge in prescribing different decoctions for treating different diseases. This has sparked my interest in herbal medicines. At the age of 9, I determined to pursue my dreams to further my study in herbal medicines. With a dream of wanting to discover the miracle healing power of medicinal plants, I landed in Professor Manaf's labs, Universiti Putra, Malaysia, to begin my research in the field of drug discovery and cancer chemoprevention in 1997. These are my mentors. From the left, Professor Manaf, Professor Nordin, Professor Nohanum, and Professor Murakami from Japan. As we are aware, cancer is the top killing disease in the world and in Malaysia. In 2012, there was 14.1 new cases of cancer, but in 2030, there will be 23.6 million new cases, which is about 67 increment. And the 5 most common cancer are lung, liver, stomach, colon, and breast. Can we cure cancer? Let us not to think of the answer now. The complexity of cancer formations limits the conventional cancer treatments and pose great challenges in cancer treatments, where only limited anti-cancer therapies are available with high efficacy for satisfactory cure rate. What do we know about cancer? Cancer is a disease of cell biology. Cells receive signal to function. Signal turn on and off the specific signaling pathway to precisely regulate cellular functions. In cancer cell, the turn on and off signal fail to function precisely due to defective DNA that contribute to the complexity of carcinogenesis. These are 6+2 cancer hallmarks describe the multiple signaling pathways found in cancer cell. This hallmark has been used as a useful conceptual framework to understand the complexity of cancer biology. Each hallmark is regulated by multiple signaling pathway. This is an example of multiple signaling pathways, mainly regulated by AKT genes. Cancer cells strive to survive and continue to grow by manipulating through these multiple signaling pathways. Just like us, we will use alternative route to escape traffic jam. Hence, what should we strategize our approach to aim at the target so that the turn on and off signal could be targeted precisely to activate and completely shut down this multiple signaling pathway and ultimately kill cancer cells. Now, I would like to share the medicinal benefits of Maslinic acids. Our research team reported the anti-tumor promoting activity of Maslinic acids for the first time in 2001, and subsequently, there are many research papers reporting the biological activity of Maslinic acids, with the highest number of publications discussing its anti-cancer properties, and followed by anti-inflammations, anti-diabetic, antimicrobial, neuroprotective, hepatoprotective, cardioprotective activities, and others, which we can see in more details from this pie chart. Thus far, there are a total of 157 studies conducted to research on its biological activities, with 29% for anti-cancer, 16% on anti-inflammations, 13% on anti-diabetic, 11% on antimicrobial, and the rest. This pie chart shows that the in vitro study of Maslic acid on various cancer cell lines, with 28% study on colorectal cancer, 11% on breast cancer, 9% on melanoma and other cancers. The types of anti-tumorigenic activities of Maslic acid is depicted in this pie chart, where 27% reporting on its anti-proliferation mechanism of actions, followed by 23% on programmed cell death via apoptosis and autophagy, and 9% on cell cycle arrest, and so on and so forth. Now, I would like to draw your attention to the 6+2 cancer hallmarks and Maslic acids. Can Maslic acid act as one compound multi-target anti-cancer drugs? Which multi-signaling pathway can be targeted by Maslic acids? This is the systematic review paper submitted by my research team for publication. In this paper, we discuss in detail on the anti-cancer activity exerted by Maslic acid via the 6+2 cancer hallmarks. So far, our research team has published several papers targeting the hallmarks. First, avoiding growth suppression. Second, avoiding immune destructions, and tumor promoting and inflammations. For the interest of today's sharing, I will only focus to discuss the anti-inflammatory effect of Maslic acid via protein kinase pathway, in which the regulatory effect of Maslic acid on protein kinase, NF-kappa B, AP1, COX-2 expressions will be discussed. The immunoblotting analysis shows that Maslic acid achieves the greatest suppressive effect on protein kinase beta-1 as compared to the rest of protein kinase isomers. For the inhibitory effects of Maslic acids on NF-kappa B activity, Maslic acid at the concentrations of 100 micromolar showed greatest inhibitory effect than oleanolic acid and slightly less effective as compared to oxalic acid. From this diagram, we can observe that Maslic acid and oxalic acid completely remove the induced AP1 binding activity at the highest concentration tested at 100 micromolar, whereas oleanolic acid partially inhibited the induced AP1 binding activity at 100 micromolar. The result obtained for the inhibitory effect on COX-2 expressions, Maslic acids and oleanolic acids suppress 50% of COX-2 expressions at 100 micromolar. Oxalic acid showed strongest inhibition effect, in which achieved 50% of COX-2 expressions at 25 micromolar. Now, I would like to move on to another study on inductions of detoxifying and antioxidative enzyme through the NRF2 ARF signaling pathway. In these studies, we determine the capability of Maslic acid in activating NRF2 expressions and translocated into the nucleus to induce antioxidative enzyme HO1 and detoxifying enzyme NQO1 expressions. As shown in this diagram, Maslic acids and oleanolic acids significantly increase nuclear NRF2 expressions. Higher NRF2 expressions was observed in Maslic acid treated cells than in oleanolic acid treated cells. Maslinic acids and oleanolic acids enhance ARE binding in a concentration-dependent manner. Effect of Maslinic acid was stronger. Binding was observed in cell treated with 25 micromolar of Maslinic acids, but not in oleanolic treated cells. For mRNA and protein expression studies, both Maslinic acid and oleanolic acid induce expressions of antioxidative enzyme HO1 in a concentration-dependent manner ranging from 12.5 to 100 micromolar. Similarly, both Maslinic acids and oleanolic acids also induce expressions of detoxifying enzyme NQO1 in a concentration-dependent manner ranging from 12.5 to 100 micromolar. We employed gene silencing method to knock out NRF2 to confirm that Maslinic acid induced both HO1 and NQO1 enzymes via NRF2 expressions. From these gel images, NRF2, NQO1, HO1 expressions were induced following Maslinic acid treatments in transfected control cells and cells transfected with scrambled siRNA. The ability of Maslinic acid to induce NRF2, NQO1, and HO1 were minimal in cells transfected with Si and NRF2-1. This confirms that NRF2 is required for Maslinic acid to induce both HO1 and NQO1 enzymes. Based on the findings presented, it suggests that Maslinic acid is a potential phytochemical which it can able to suppress pro-inflammations via protein kinase pathway and activate antioxidant response via NRF2 ARE pathway. From the current study, we would like to support the citations proposed by Zhong et al. In 2016. He stated that cancer treatment for the future would be based on the drug combinations that target at several molecular alterations or cancer hallmarks. This is challenging, but it is the promising precision cancer treatment. Thus, it is hoped that Maslinic acid could be further developed as a multi-target anti-drugs. Before ending my sharing today, I would like to thank my grandmother and mother for instilling my interest in herbal medicines. Also, I would like to thank Professor Murakami to provide the opportunity for me to have research attachment at his lab and successfully isolated Maslinic acid, as well as to transfer the anti-tumor promoting acid back to Malaysia. With that, I thank you for your kind attention.